Hello everyone, welcome to my series in which I talk about the book Principles of Anatomy and Physiology. So today we're covering chapter 3 of the book, which is the cellular level of organization. So as always, let's start with the mind map. So today we're firstly going to talk about the three main parts of a cell. Then we're going to dive into each of them and explore them in some detail. We're going to cover the structure, the function, as well as some properties of the plasma membrane. We're going to talk about how cells transport things or move things across our plasma membrane. There are three main mechanisms. We're going to talk about the cytoplasm, which is the cytosol, plus a lot of organelles in it, and we're going to briefly talk about some of the organelles. Then we move on talking about the nucleus, so some of its key structures as well as its function. Then we're going to talk about some important processes that are happening in our cell, which are protein synthesis involving two main steps, cell divisions. We're going to distinguish between somatic cells and reproductive cells. And we're also going to talk about aging and different ways of cell death. We start with talking about the three main parts of a cell. So I'm sure this must resonate with you a lot from biology class. Firstly, we have our plasma membrane, which is the wall of the cell, so it separates the internal environment from the external environment. Then we have our cytoplasm, which is the cytosol, which is the fluid part, plus the organelles. So organelles are like tiny machinery that perform specific functions, and together all the organelles allow the cell to complete all the things it needs to do. So we're going to talk about a lot of organelles in the cell later, as well as cover their functions, so stay with me. Then we have our nucleus. So our nucleus is actually also an organelle, but we have listed it here separately because it is really important. It houses the genetic information of us human beings, or also DNAs. So we'll also talk about this in a lot of detail later. So first of all, let's talk about the plasma membrane. So the main word describing the structure of the plasma membrane is the fluid mosaic model. So let's break this word down. So fluid, it means moving around. So in the molecular arrangement of our plasma membrane, the lipids, they're not sealed so tightly. So it's not like a steel wall, but it's like something that's sort of liquidy that can wobble around. And it is called a mosaic because there are a lot of different proteins embedded in the cell membrane. So as we can see here, these two tadpole-like structures on each side, these are making up our plasma membrane, and it's not structured so tightly, and there are spaces for different proteins that we see. So some examples are the glycoproteins, the peripheral proteins, And then we have our phospholipid bilayer, which is also a very important thing to talk about when we talk about plasma membranes. So, as I mentioned, we have these two tadpole things that have like two tails. So they have a blue head and two tails beneath them. These are phospholipids. So as you might recall from when we talked about the last chapter, these are molecules that have a hydrophilic head, which likes water, and a hydrophobic tail, which does not like water. So this arrangement is really logical because the two parts that like water of the phospholipids, the heads, they're facing outwards. They're facing the solutions, the extracellular solutions as well as the intracellular solutions. And then the parts that don't like water, the hydrophobic fatty acid tails, they're facing toward each other, so they're not making contact with any fluids or liquids in that matter. So this is why we have a phospholipid bilayer or lipid bilayer. We have two layers of phospholipids facing tail towards tails. We can also see there are some other components in our lipid bilayer. We have, for example, we have cholesterol, you can see here. We also have some lipids, some other lipids. So these are dispersed between our lipid layers, and the way they stick out also depend on their polarity. So if it's a nonpolar molecule, it tends to like hide in the middle of this bilayer, but if it has a more polar side, it might stick out of the membrane in one side. Then we have a lot of membrane proteins, as you can read here. First of all, let's distinguish between integral and peripheral proteins. So they are classified also based on their location in our lipid bilayer. So integral proteins, they extend into or through the lipid bilayer, and they are firmly embedded. So these are mainly transmembrane proteins, meaning that they span the entire length of the lipid bilayer, and they protrude into both the extracellular fluid as well as the intracellular fluid, which is basically the cytosol. So they go through the whole bilayer, so they're called integral or transmembrane sometimes. And peripheral proteins, from the name, we can understand that they are not so firmly embedded in our membranes, and usually they're attached to the polar heads of membrane lipids or to integral proteins at the inner or outer surfaces of the membrane. And then we also have, so for example, an example of an integral protein that we can see here is a glycoprotein. So glycoproteins are proteins with carbohydrates attached to the ends, and they protrude into the extracellular fluids. So these are some examples. So what do membrane proteins do? There are a lot of things that membrane proteins can do. For example, we have ion channels. So they form a pore, and through this pore, ions can flow through this membrane. So if we didn't have such pores, then ions, they would not be able to cross the membrane, because in the hydrophobic part of our lipid bilayer, the ions are strongly repelled because of their charge. So ion proteins, which are integral membrane proteins, are a way of allowing ions to pass through, to cross through our membrane. We have carriers, which transport different substances also across the membrane through changing their shape. We also have receptors. Receptors are also integral proteins. So they recognize specific ligands, and they react in a certain way that alters the cell's function. For example, maybe hormones can bind to our receptors that change something in the cell. We can also have enzymes. We can have linkers that anchor some filaments. We can also have glycoproteins that function as the identity marker of the cells. So now, let's talk about some of the main functions of our plasma membrane. So the membrane has 4 main functions. So sorry for the little technical difficulty that we had there with the recording, but let's continue with where we left off about the functions of the plasma membrane. So there are 4 main functions of the plasma membrane. Firstly, it acts as a barrier that separates the inside as well as the outside of the cell, as we already mentioned. Secondly, it is able to control the flow of substances into and out of the cell. So it is acting like a gate for whatever it comes in. It will check whether this is appropriate for the cell, is this something harmful. So it prevents too much of toxic things of going into the cell, and it also controls the concentrations, etc. It also controls what goes out of the cell. So it might expel some waste out of the cell through the plasma membrane. Thirdly, it helps to identify the cell to different cells. So as we talked about, glycoproteins, they serve as an identity marker of the cell. So there are also things in the immune system. So these proteins, they will hold out identities of the cell itself. So immune cells can check whether these cells are healthy or not. And last, they also participate in intracellular signaling, so signals between cells. This is also something that we'll talk about when we mention the nervous system. There are a few properties of the plasma membrane that allows us to do these things. Firstly is what we call selective permeability. So permeable meaning allowing something to pass through it. So impermeable means something cannot pass through it. So by selective permeability, we mean that there are certain substances that can pass through the plasma membrane more readily than others. So some molecules that are really small and molecules that are relatively nonpolar, they are really permeable across the membranes. And these can be, some examples can be oxygen molecules, carbon dioxides, and also small steroids. Water also, because it is a really small molecule. And then we also have some impermeable molecules. These really large carbohydrates or really polar molecules like ion cannot pass through. So water is special, even though water is polar, it is so small that it can squeeze through our lipid bilayer. We also have something called the electrochemical gradient. So basically, this refers to the difference in electrical charge between the two sides of the plasma membrane. So usually the inside of the cell is more negatively charged than the outside of the cell. This also plays a key role in producing action potential that we'll talk about when we talk about the nervous system. So these are some of the main things you need to know about the plasma membrane. Moving on, let's talk about transport across the plasma membrane. So, as we talked about, the plasma membrane is a barrier that surrounds the whole cytoplasm, that surrounds the whole cell and keeps it separate from the outside environment. So how are molecules supposed to move through? Because a cell cannot sustain itself. It needs nutrients, it needs oxygen from the blood, and it also has waste that accumulates that it needs to expel. And other than that, it also needs to perform functions. It needs different signals. It needs to pass out signals. So how do cells manage to do this? So we're going to divide this into two main types of processes. We have passive processes. These are processes that do not require energy, so they can be performed by themselves. You do not need to push these reactions to happen. And they go from higher concentrations to lower concentrations. So this should be quite easy to understand. For example, if we mix two solutions, one has a high concentration of, for example, salt. It tastes very salty, and the other solution has a low concentration of salt. It's not that salty. Maybe you can't even taste it. And when you mix it together, diffusion actually happens. So the part of the solution with more salt goes toward the part of the solution with less salt. So it naturally goes from a higher concentration to a lower concentration. And overall, the solution is, the saltiness of the solution is in between that of the two that we have mixed. So this is the overall idea of passive processes. We have, first of all, simple diffusion. This is what I just mentioned, but in this case, we have a membrane separating it. So some of the factors that can affect this transfer includes, first of all, how steep is the concentration gradient. So if there is a greater difference between the concentration between the two sides of the cell membrane, then they diffuse more fastly. This is quite intuitive, I think. Secondly, we have temperature. So the higher the temperature, the faster the rate of diffusion. So if we think about the molecular model of things that you might have learned about in chemistry, faster temperatures mean more kinetic energy. They means they're vibrating more, they're moving around more. So this means they move around more, they pass the membrane more, then they balance out more. So they reach an equilibrium state faster. The surface area also matters. So a larger surface area means a faster diffusion. Then also the diffusion distance matters a lot. So the greater the distance, the longer it will take. So both the surface area and the diffusion distance are two things that play key roles when we think about breathing. So simple diffusion happens when a substance can move freely across the plasma membrane. And these are usually, as we have mentioned, very permeable things like oxygen, carbon dioxide, some other gases, some steroids, also some vitamins. And this also happens with water and small molecules like urea, like alcohol sometimes. Then we have facilitated diffusion. So this is when the solute is too polar or too charged or too large to move through by simple diffusion. And it is helped by a protein through a process called passive diffusion. So there are different types of facilitated diffusions. So what basically happens is there is a protein that helps our solute cross the plasma membrane in some way. In a channel-mediated facilitated diffusion, there's basically a channel, which is quite intuitive, in which our solute can flow through. We can also have carrier-mediated diffusions. So think of carriers as V-shaped things, and they can click towards one side and click back toward the other sides. So when it's toward this side, then there is some solute molecules can come and attach here, which activates our carrier, and it switch to the other side. And our molecule, which is originally attached here, it can then come out and leave. And through this process, it is able to diffuse, but with the help of some proteins. So this is really similar to simple diffusion. The only difference is due to the selective permeability of our plasma membrane, our solute needs a protein to help it cross the membrane. Then we have osmosis. So this might seem an intimidating concept, but don't worry. Osmosis is basically the diffusion of water. So we talked about how plasma membranes are selectively permeable. This means that there are substances that are not able to pass the membrane, but water can pass the membrane. So when we think about the relative concentrations of water and of, for example, some large carbohydrate that cannot pass the plasma membrane, the water is able to diffuse, so the concentration of water can change, but the concentration of the other solute cannot change. So when thinking about osmosis, think about the relative concentration of water. So if the concentration of a solute is high, then the relative concentration of water is comparably lower. And to the other side, side has a low concentration of our solute, so comparably it has a higher relative concentration of water. So we know that water can move freely across our plasma membrane, so the side with the higher relative concentration of water will move to the side with a lower relative concentration of water. And this is basically the concept behind osmosis. And water can also move faster through special aquaporins, which are integral membrane proteins, again, that functions as water channels. So when we talk about osmosis, we talk about something called tonicity. So tonicity is basically a measure of the solution's ability to change the volume of the cell by altering the water content. So in an isotonic solution, the concentration of solutes across the cell membrane, across the plasma membrane is the same. So outside same, inside the same. So the water is in equilibrium. It would not want to move. So this is in the case of, for example, red blood cells. And this is also why when we do IV injections, we use a 0.9% sodium chloride solution, just to, we call it a saline solution because this is the normal concentration of salt in our blood. So this would not cause cells in our body to swell or burst or, Then we have hypotonic solutions. So these are solutions where there are lower concentrations of solutes than the cytosol inside the cells. So think of hypo as in low. So there is a low concentration of solutes. We think about it as outside of the cell. So in this case, the water molecules will want to enter because, as we talked about, a low concentration of solute means a high relative concentration of water. So this is outside the cell. Inside the cell, we have a relatively lower concentration of water. So the water wants to enter from outside of the cell into inside of the cell. So this causes the cell to swell and eventually burst. So this process is called lysis, which is basically bursting. This is actually something that we can see in daily practices. Farmers sometimes spray water onto their crops. This is entirely healthy, but through this hypotonic solution, because the water does not have a lot of solutes compared to the cell. So in this type of hypotonic solutions, water will want to enter the fruits and plants and make them look more fresh, make them look more big. Then we have hypertonic solutions. So think of hypertonic as in hypertonic. So this means there is a higher concentration of solute outside of the cell. So this means a lower relative concentration of water outside of the cell. So the water will want to go from high to low, which is from inside of the cell out of the cell. So water leaves. And this shrinking is called crenation. So we can see this in pickled food. So in pickled food, basically we put food in really salty water and we leave it and we take it out and let it dry. And then we see this really cringe and crumple up food of whatever we're pickling. So this is the process that happens behind pickled food. So these are all passive processes that we see. Next, let's talk about active processes. So everything is the opposite of passive processes. Active processes, as their name suggests, require energy. So you're actively pushing one thing to the other side of the plasma membrane. And this is going against the concentration gradient. So originally we're going from high to low and we balance things out. And now consider one high, one low. We're pushing this gap further apart. So we're going from low to high. So quite intuitively, this is a process that goes against what would naturally happen. And this is also a reason why it requires energy. We usually divide active processes into primary and secondary active processes. So in primary active transports, energy derived from the hydrolysis of ATP changes the shape of a carrier protein. So the carrier protein actively pumps a substance across the plasma membrane against its concentration. For example, think about ions like sodium ions, for example. We have the sodium-potassium pump that will also cover again in the nervous system. So an enzyme called basically the sodium-potassium pump acts as ATPase, and an enzyme which is an enzyme that hydrolyzes ATP. So basically it pushes the sodium as well as the potassium ions against their concentrations, concentration gradients, and accumulates a lot of sodium on one side and a lot of potassium ions on the other side. So this also contributes to an electrochemical gradient, so there is a buildup of charge. So it is using energy to actively change the shape and pump these ions to the other side of the membrane. Then we have secondary active transport. So you can think of secondary active transport as basically riding on another person's car for free. So for example, the energy stored in gradients, in chemical gradients, for example, of hydrogen ions, they are used to drive other substances across the membrane against their own concentration gradients. So because our sodium or hydrogen gradients, they're previously established by primary active transport, our secondary active transport is indirectly using the energy obtained from the hydrolysis of ATP. So they're not directly consuming energy, but they're basically free riding on the energy that was previously used. So now let's talk about the last type of transport. we're not talking about this in terms of energy, but this is special and really easy to remember because we have something called a vesicle. So vesicles sound sort of like vehicle, right? So vesicles are basically small sacs, and they are like vehicles that transport substances into or out of the cell. We have endocytosis, in which materials move into the cell in a vesicle, and the vesicle fuses with our plasma membrane, and after this fusing, whatever is in our vesicle gets into our cytoplasm. So remember endo as going in and exo as going out. So in exocytosis, materials move out of a cell. And both processes require supplies of ATP, and they rely on the plasma membrane, in that they fuse with the plasma membrane, or they basically boggle out of the plasma membrane. So some useful examples are, for example, phagocytosis, in which, for example, a white blood cell engulfs another cell, for example, some bacteria or some yeast cells. Some cells, actually a lot of cells carry out bulk phase endocytosis, which is also called penocytosis. This is like the drinking of the cell. Phagocytosis is cell eating, and endocytosis is the drinking of the cell, so really humanized. So this is where endocytosis takes in small droplets of extracellular fluid. We also have what is called transcytosis. So in transcytosis, vesicles may be used successively to move a substance into, across, and out of the cell. So it's a combination of both processes. It goes in, do some processes, and fuses with the plasma membrane and goes out again. So it's trans, across. So these are some of the main ways that we usually move molecules across the plasma membrane. Now let's talk about the cytoplasm. So the cytoplasm is matter that consists of all the cellular content between the plasma membrane and the nucleus. And there are two main components of our cytoplasm, which is the cytosol, which is the fluid portion of the cytoplasm. So it is mainly water, which constitutes between 75% to 90% of the cytosol mass. And it also has a lot of dissolved or suspended components. Some types include ions, glucose, amino acids, etc. And then the cytosol is also the main site for chemical reactions required for a cell's survival or existence. So in the cytosol, enzymes can catalyze a process called glycolysis, which is part of the process of cellular respiration, in which we generate ATP. We also have what is called the cytoskeleton. So these are some protein filaments inside our plasma membrane that support the cell. These are like the iron beams in the building that supports the building together. So there are three main types. First of all, we have microfilaments. So microfilaments are thin elements. They're the thinnest elements in the cytoskeleton, and they're mainly composed of the proteins actin and myosin. So they're prevalent at the edges of a cell, and their main function is to generate movement as well as provide mechanical support. So with movement, it is responsible in actions such as muscle contraction, cell division, some locomotion such as the migration of some embryonic cells during development. And then mechanical support just basically means it provides basic strength and holds the shape of the cell together. And they also anchor the cytoskeleton to some proteins that are in our plasma membrane that we mentioned. They also provide support for what we call microvilli, which are tiny projections outside of the cell that are like tufts of hair. Next, we have our intermediate filaments. So they are thicker than microfilaments, but they're thinner than microtubules. They're intermediate in their thickness. They can be composed of different proteins, and they're mainly strong. They form strong bonds, and they're found in parts of the cell which are subjects of stress or pressure. And they mainly help stabilize the position of different organelles, for example, the nucleus, and they help the cells attach to each other. Lastly, we have our microtubules. So these are the largest components of the cytoskeleton. They're long, hollow tubes, and they do not branch, unlike the first two. And they are assembled in structures called centrosomes, which we'll talk about really shortly. They grow outwards from the centrosomes towards the periphery of the cells, and they help determine the cell shape. They're also important during cell division, and we'll talk about these really shortly. Now, in the cytoplasm, beside the cytosol, we also have suspended organelles. So, as I mentioned, organelles are these small specialized structures that have their own shapes, their own functions. So let's talk about some of the organelles that we usually encounter in our bodies. We have our centrosome, which we mentioned. It is the center of organizing our microtubules. So we usually have two centrioles. So centrosomes comes in pairs of centrioles. The two centrioles are cylindrical structures, and they are a cluster of there are 9 clusters of trios. So we have trios, which are 3 microtubules, triplets, in a circular pattern, and they are in 9 triplets. So like it forms like a ring of triplets. And so centrosomes are essential during cell division. They replicate, and these microtubules, they pull our chromosomes apart. So this process of cell division will be shown later in this chapter. Next, we have cilia and flagella. So microtubules are also the dominant components of these two. These are, so let's distinguish between cilia and flagella. So cilia, they're mainly short. They're numerous in numbers. So often in a cell, you can see like maybe hundreds of cilia projecting from the cell surface. And they're also arranged in clusters. They have 9 clusters of doublets, which are not triplets compared to our centrosomes. And they're anchored to a basal body. So the basal body is below the plasma membrane. And they're similar in structure to centrioles. So in cilia, they display a oar-like pattern. So we think of them like oar. They have this pattern of, it's not exactly like whipping, but they float around, and they're really fluid structures. You can think of them like, so just basically think of it like oars digging into water. And the coordinated movement of many cilia in the surface of a cell can cause steady movements of fluids, so they mainly move fluids across the surface of a cell. They're present in cells such as in the respiratory tract, so they need to sweep out foreign particles that we might breathe in. And then we have our flagella. So flagella, mainly think of it as a long whip. So it whips around, it's much longer, and they're so long and powerful that they're able to move an entire cell. So the only example of a flagella that is present in the human body is sperm. The tail of the sperm cell is a flagella. But flagellas are relatively common in bacteria, but we don't have a lot of them. Moving on to our next organelle, these are in alphabetical order. So we have our ribosomes. Oh, I'm sorry, these are actually not in strict alphabetical order, but sort of. So ribosomes are the sites of protein synthesis. We'll talk about the process of protein synthesis in much detail later. But ribosomes are tiny structures, and they're mainly made up of ribosomal RNA. They also include different proteins, and we usually think of ribosomes as two subunits, one above the other, and they sort of cling together, and there's like this area in the center that allows proteins to go through them. So I'll have some graphical details for this later when we talk about protein synthesis, but they're really important in terms of making proteins. Ribosomes can either float freely in the cytoplasm, or they can be attached to endoplasmic reticulum. So the endoplasmic reticulum is a network of membranes. They're sac-like, and they extend from our nucleus. It can be connected to the nucleus and project into the cytoplasm. It is really extensive, and it has like a membrane of its own. It consists a large portion of the membranous area in the whole cell. And we have two forms of ER, as you must know. We have rough ER. So rough ER is directly continuous with the nucleus. It's rough because there are a lot of ribosomes attached to the surface of the rough endoplasmic reticulum. And some of the molecules that they mainly make can include, so they're also basically places for making proteins. Because there are a lot of ribosomes attached, and these proteins can attach. They can enter into the rough ER, into like the folded spaces, where they're processed and stored and sent to different places they're needed. And they can molecules like glycoproteins or phospholipids may be incorporated into the things that are made. And they transport proteins via vesicles, which is exocytosis, as we talked about in the cellular transport section. Now, smooth ER is different from rough ER because it does not have ribosomes attached on the outer surface, but it has unique enzymes that makes it functionally more diverse. It can make different things. It mainly synthesizes fatty acids and steroids. It can also perform the process of detoxification of some soluble and harmful substances. Then we have our Golgi complex. So after all our different molecules, our proteins and fatty acids are made, they need to be sent to the places where they're needed. They need to be sorted. So the Golgi complex is basically a center of processing and sorting and packaging and shipping out these different proteins and things. So think of the Golgi complex as a shipping center. It is really close to our ER, and it has two phases. It has the cis phase, which is entry, and it has the trans phase, which is exit. And also it is sac-like, so we just see a lot of sacs connected to each other and some vesicles that are going in and going out. Then we have our lysosomes. So lysosomes, as you might see from the word lyso, like you can think of it as lysis, so it's dissolving things, it's breaking down things, so it contains a lot of enzymes at really acidic pHs, which is able to dissolve a lot of things. And they also move some of the final projects of digestion. Sometimes when entire organelles are worn out, they're no longer functional, they are digested. They're engulfed by our lysosomes in a process called autophagy. And sometimes lysosomal enzyme, like the enzyme released from these lysosomes, they can destroy the entire cell, which is called autolysis, and it is a process of destruction of the entire cell, which we'll also discuss a little bit later. Then we have our peroxisomes. So peroxisomes are similar to our lysosomes, but they're smaller. So both of these are small spheres with a membrane. So peroxisomes have enzymes that can oxidize a lot of organic substances. For example, amino acids and fatty acids can be oxidized in peroxisomes as part of metabolism. And they can also organize some toxic substances such as alcohols. And they are really abundant in our liver, especially for the detoxification process. Then we have our proteasome. So, as we just mentioned, lysosomes degrade proteins and deliver them to vesicles. And the continuous destruction of unneeded, damaged, or faulty proteins is the function of proteasomes. So they continuously degrade proteins after they have completed they have accomplished their function. So these proteins are destructed, and they're broken down back into their basic parts. So they might be like these basic building blocks can be recycled, perhaps. They're also involved in some diseases in which the failure of proteasomes to degrade abnormal proteins can lead to clumps of misfolded proteins accumulating in the brain cells of people, and this can lead to Parkinson. So this is a really important organelle in terms of cleaning up the cell and making sure that there is no unnecessary debris or waste. Next, we have our mitochondria. So we all know that the mitochondria is the powerhouse of the cell. They generate most of the ATP through aerobic respiration. They have two membranes. They have an external membrane and an internal mitochondrial membrane. Inside, we have the matrix, which is the space, and we have cristae, which are folds of the membrane inside. So we have this sort of ragged structure with the membrane folded in a lot of places. So we have our mitochondrial matrix, which is enclosed by the internal membrane, and as we mentioned, they're really important in producing ATP, which is the main energy currency of our body. So we'll talk about the specific process of generating energy when we talk about some muscles and when we talk about energy in general. Sorry. So these are some of the main organelles of the body. And moving on, let's talk about the nucleus. So as I mentioned before, the nucleus is also an organelle of the body. However, we're talking about this separately because it is really important in terms of storing and passing genetic information. So first of all, the nucleus is enveloped by a nuclear envelope. So it's a double layer. It's an envelope because envelopes have two layers and we have things in between, right? So it's basically really similar to our phospholipid bilayer membrane, but it also has two layers folded together. And in these nuclear envelope in our membrane, we have nuclear pores. So pores are a circular arrangement of proteins around a central canal, and they control the movement of substances, some molecules, some ions. Then we have a structure called the nucleoli, which is inside the nucleus. So they're spherical bodies that function in producing ribosomes. So as we talked about before, ribosomes, they produce proteins, but what makes ribosomes? Ribosomes, they have, as we mentioned, they are mainly made of RNA. And because this is related to genetic information, then our nucleus plays a certain role. So the nucleoli makes these ribosomes, and each each nucleolus is basically a cluster of proteins, DNAs, and RNAs. And they synthesize our RNA, which then fold and with proteins make up our ribosomal subunits. Then we have our chromosomes and chromatin. So genes are arranged in chromosomes. As you know, human somatic cells or body cells, we have 46 chromosomes, 23 chromosomes from each parent. And we have our chromatin, which is basically this complex of DNA, proteins, and some RNA. So all of this DNA that's coiled together or tangled, that's called the chromatin. And the total genetic information carried in a cell or in an organism is called our genome. So our DNA, we know that we usually visualize our DNA in X shape. So in our DNA, in our chromosomes, we have DNA folded together around proteins that are called histones. And these, just think of DNA being wrapped around, like line over a ball around proteins that are called histones. And then these histones assemble together, and these histones then wrap around again, and they form this sort of X shape. And yeah, this is basically how we have this characteristic shape of a chromosome. And after DNA has replicated, it can form two chromatids. So in an X, we have two legs, right? These are sister chromatids, and they're identical to each other. And we'll explain why we need chromatids when we talk about mitosis and meiosis. So why is the nucleus so important? So basically, as we've mentioned, the nucleus, the nucleus is the site that holds all of our DNA. The DNA never leaves our nucleus. Keep in mind that the DNA never leaves the nucleus. So when we talk about protein synthesis, it's always the DNA being converted into other forms, such as RNA, and then the RNA going out of the nucleus and transmitting this message. So our genetic information is really protected inside the nucleus, which is the core. And without the nucleus, we do not have the genetic code which defines us as us. Now let's talk about the process of protein synthesis, which is one of the main processes that are carried out in the cell. So protein synthesis is divided into two stages. First, we have transcription, which is the process of DNA converted into RNA, and then we have translation, in which messenger RNAs are converted to proteins. So how to remember the difference? So how should I not confuse these two processes? So we all know what a transcript is. So think of the DNA as the person that is talking, the person giving all the information, and think of RNA as an intermediary. So the RNA is listening carefully, it is noting down what the DNA said, it is writing down the orders, recording the information in a really precise way. So this is transcripting. And then translation. Translation, we usually think about it in terms of one language going to another language. So they have fundamentally different forms. They're different languages, right? So in protein synthesis, this change in form, think of it as going from genetic code from the ACTGU into the amino acids that are encoding in protein. So we're also going a change in language in the cell. We're going from genetic codes, nucleotides, into amino acids and proteins. So this is translation. Now let's look at the two processes specifically. First, we have transcription. So in transcription, DNA is converted into messenger RNA, and this is mainly carried out by our RNA polymerase. So our DNA goes through the RNA polymerase, and there are segments of DNA where transcription begins, and they are special sequences that are called promoters. So when the RNA polymerase sees a promoter, it knows, oh yeah, I have to start listening, the transcription needs to start, right? And then we make RNA in a complementary manner. So we know that in DNA, A and T pair up with each other, and G and C pair up with each other. In RNA is really similar, but instead of A, we have U. So whenever we see an A on DNA, we have U in the RNA. When we have T, we have corresponding A. G with C, C with G. So we're transcripting this in a complementary manner. Then we also have terminators, which are special nucleotide sequences, which tells the RNA polymerase, hey, this is the end of the sequence. You do not need to transcript anymore. So we have our RNA sequence, and this transcript immediately after transcription is called pre-mRNA, pre-messenger RNA, and it can exit the nucleus through our nuclear port, as we mentioned before. So there's also a process that is called splicing that is performed. So why is it called pre-messenger RNA? Why do we have this pre? This is because the messenger RNA is processed. So in the process of transcription, what is in the DNA is exactly converted into RNA. But there are pieces of DNA that are not needed to code for proteins. So there are sections of DNA that are not coding for proteins. This is also transcripted into RNA, but when we're trying to make proteins, when our ultimate goal is to make proteins, we do not need these sections. So we would slice these sections out. So regions on the RNA that are not coding for parts of a protein are called introns. They are snipped, deleted, they're sliced out by what we call small nuclear ribonucleoproteins, which are the green structure, the S and R and P that you see on the diagram. And the exons, which do code the proteins, they are left together. Then we have our translation process. So we already have all the instructions we need in genetic code. Now we need to convert it into a protein. So we see here our ribosome. So the purple and the sort of red and pinkish parts, they are the subunits of ribosomes that we talked about. And as I said, they cling together, and there is the space in between that allows RNA to go through them. So first, we need to introduce the concept of codons and anticodons. So when we talk about protein synthesis, when we make proteins, a pair of three nucleotides, they correspond to an amino acid. So for example, our start codon, AUG, this corresponds to the protein methionine. So basically read the mRNA in triplets. They come in three. For example, in this graph, we have AUG, UGC, CUG. Think of it in units of 3, 3, and 3. So these are called basically what we say codons. So first of all, our ribosome looks for a start. It looks for a start codon, which is AUG, as I mentioned. The corresponding tRNA, the UAC, is called the anticodon. So you see here these blue sort of tangled structures. These are our transfer RNAs. They are our tRNAs. So basically, we see that they carry two main things. Firstly, they have nucleotides on them, and then they have this sort of line that connects them with this one amino acid on the top. So these they identify codons. So when we see, how does the ribosome recognize a AUG start codon? This is because the anticodon, the UAC carried by the tRNA, is able to match, is able to stick to our AUG starting codon. So this gives us our first our first amino acid, And after we've read the first three codons, we move on to the next three, which in this case, we have UGC, I believe. So we see that there are three sort of impressions, there are three spaces in the ribosomes. So they're called the E site, the P site, and the A site. So the P site is the peptidyl site. It binds to the RNA that's carrying this peptide chain. So the chain is always on the P side. The A side, the amino acid side, is the place for entering. So a new tRNA comes in. So in this case, the tRNA with the anticodon AUC, it comes in. It attaches to the UGC on our messenger RNA, and then it moves on. So we see the methionine on the starting codon. It moves, it attaches to our second. It attaches to the new tRNA in the amino acid chain and moves on to the A site. But then it exits. So our first tRNA exits out of the E site, which stands for exit site. And our original, so like basically the actually what I said was a little imprecise. It's not the tRNAs that are moving around, it's actually the whole RNA chain moved forward. So the tRNAs are still binded to the codons, so the anticodons are always connected to our codons. But the mRNA, it moves forward in the ribosome. So when the tRNA reaches the exit site, and it basically exits. So it is released from the ribosome, and the tRNA that is previously at our A site, it is now at the P site. Basically, the whole RNA shifts forward by 3 codons. By 1 codon, or 3 triplets, I'm sorry. And this process continues again and again until we have the last, when we reach like an ending codon. So there are 3 ending codons. These codons, their tRNAs do not bring an amino acid. So we have the 3 codons U go away, which is UAA. Wait, let me think about this. Yeah, we have our 3 anticodons. They are U go away, UGA, U are away, UAA, and then U are gone, UAG. So this is indeed sometimes hard to remember, and I forget it a lot of times. Try to use the mnemonic that I just said. So think of the process of a person going away. So first, you go away, UGA, you are away, UAA, then you are gone, UAG. So think of it as the process of a person leaving you, right? So with these stop codons, no amino acids are brought. So basically, the amino acid chain is then released from the ribosome. And then in our amino acid chain starts to fold, as we talked about in chapter 2, the chemical level of organization. We have primary, secondary, tertiary, and quaternary structures that our proteins can fold into. And then this basically concludes the process of protein synthesis. We have made a protein from coding DNA into processed messenger RNA, and with the help of transfer RNAs, we converted them into a sequence of amino acids, which then fold naturally into protein. So now let's talk about the 2 types of cell divisions. So these are key mechanisms that allow cells to basically grow in size, replicate in number, and also produce the reproductive cells that are essential for creating offspring. So first, let's look at somatic cell division, which is the division of our body cells. We have what is called the cell cycle. Firstly, a cell is normally an interphase. So most of our cells are going to be interphase. So these are periods in which the cell grows in size, it replicates organelles, it replicates the DNA, replicates other things like cytosolic components, centrosomes. So this ensures that the daughter cells have identical copies of both things that they need. Then we enter into mitosis. So think of mitosis, think of the 4 phases of mitosis as in PMAT, P-M-A-T, or there's another mnemonic that says Professor met Anna at the telephone. So P-M-A-T. Firstly, we have our prophase. So in prophase, 2 things appear and 2 things disappear. So first, our nucleus disappeared. Then our nucleolus, our nucleoli, as we talked about, they also disappeared. So what appears are firstly spindle fibers which are basically microtubules from the centromeres, centrosomes, and then chromosomes also appear. So chromosomes, they condense into our X-shaped form, and also the chromosomes here are already replicated. So the X shape, as we mentioned, there are two identical sister chromatids, so there are two copies of the same genetic information, and each daughter cell gets one copy. Then PMAP, we arrive at the metaphase. So in the metaphase, the chromosomes, they line up in the center. We have this sort of line in the center, and yeah, this is basically what happens. We have this this line where they line up is called the metaphase plate. Then we have the anaphase. So in anaphase, the centrosomes, they split into two parts. So what has originally been connecting the centrosomes in the center, the centromere, they're basically pulled apart. The spindle fibers, they have attached to the centromeres. They pull the two sister chromosomes of a chromosome apart into the two poles, the two ends of a cell. And then in telophase, the movement of our chromosomes stop. So they're already pulled to the respective ends of the cells, and a nuclear envelope is starting to form. Our nucleoli is reappearing, and then our DNA, originally in its condensed form in chromosomes, it starts to dissociate. It starts to loosen up into basically this. We can see these lines and masses of basically DNA. So here also 2 appears and 2 disappears. This is exactly the opposite of prophase. So our nucleoli appears, our nucleus disappears, but our chromosomes are disappearing, and eventually our spindle fibers also disappear. So this is the process. So mitosis is a process of dividing the chromosomes, dividing the sister chromosomes equally. And then we have the cytokinesis. So the cytokinesis is a process in which the cytoplasm divides. So we can see here a cleavage furrow being formed. So this is an indentation in the plasma membrane that is somewhere during the telophase or a little after the telophase. And it appears midway between the centrosomes and extends around the whole cell. So think of it as basically a cylinder in the middle being clinched together, and eventually this contractile ring, it moves inwards and inverse. So what is originally like, maybe this is the cylinder, we have our cleavage furrow. Oh my god, that's hard. So we have our cleavage furrow, and it moves smaller and smaller. It's like a sphincter. Originally it basically it pulls so inwards that the 2 cells break apart, and the cytoplasm is divided between these 2 new identical cells. So in this process, we made 2 identical cells, and these cells then continue on to interphase. So this is the cell cycle in which the cells grow and divide. However, sometimes the cells go into a phase that's called the G0 phase. So this is when there are signals. That tells the cell to exit the cell cycle. So it goes into a stagnant state where it no longer moves, it no longer divides. It just stays there. So yeah, that's called the G0 phase. Let's talk about the division of some reproductive cells. So this is usually called meiosis. We start out with a diploid cell. We started with also just basically a duplicated cell, and then we divide 2 times. So we have meiosis 1 and we have meiosis 2. We divide 2 times and we end up with haploid cells. So we'll explain this. So in humans, we know we have 23 pairs of chromosomes, or we have 46 chromosomes in total. And as I mentioned really briefly, 23 chromosomes come from each parent. So for each pair of chromosome, one is from your father, one is from your mother. So in the process of meiosis, we for each pair of chromosome, only one chromosome is needed, because you're giving this to your baby, right? You only give your baby one chromosome, and your partner gives your baby the other chromosome for each pair. So this is why we divide 2 times. So first, we have our replicated, duplicated chromosomes. In meiosis specifically, we go through a process of crossing over, in which chromosomes, they undergo we can see basically their legs, sometimes they overlap, they exchange parts. So this is a genetic recombination that boosts the diversity in genes. And then after the crossing over, we grow through the same PMAT phases. In the metaphase, they line up. In the anaphase, they're separated, and in the telophase, they're pulled apart. But here we're not pulling apart sister chromatids, we're pulling chromosomes. So you can see that 2 chromosomes are lining up together in our metaphase. So this is because the pair that is lining up together is the pair of homologous chromosomes. So they code for the same characters, they code for the same traits, but they might have different genes. So basically think of them as identical in size, and the genes inside these chromosomes, they determine the same thing. So they might, both of these code for your hair color or your eye color or something like that. And the difference is that they carry different genes, which give you different traits. So one of the chromosomes might give you brown hair, and one might give you black hair. So they're coding for the same traits. They have identical structures, they have the same sizes, but they can differ in what traits they carry. So our pair of chromosomes line up, and then they're pulled apart. So this is the process in which we go from a diploid into a haploid. So originally you have 2 chromosomes for each pair of chromosome. After pulling them apart, you only have 1 chromosome for each pair. So we go from 2n into n. And then this is the first stage of meiosis, where we separate homologous chromosomes. In the second meiosis stage, basically we separate sister chromatids. The sister chromatids are no longer exactly identical due to crossing over, but think of them as identical, and basically it's just the two duplicated portions of a same or sort of same chromosome being pulled apart. So in the whole meiosis 2 process, you're still a haploid cell, but you're going from a duplicated piece of DNA into an unduplicated piece of DNA. So the PMAT stages are still exactly the same. So this is basically meiosis. I think I have a chart that compares meiosis and mitosis. Let me see if I have. Unfortunately, I do not have the graph of a comparison of mitosis and meiosis with me. But basically the difference is that in mitosis, or the division of somatic cells, the body cells, we maintain the exactly same genetic information. We're duplicating and then pulling it into 2 copies. But in meiosis, we have crossing over, we have genetic diversity. We have, we pulled apart not only duplicated portions, so we replicated our DNAs, and we pulled apart not just the DNA into 2 copies, but we pulled it into 4 copies. And each copy no longer has pairs of chromosomes, they only have 1 chromosome for each pair of chromosomes. So then this one chromosome combines with the other 1 pair of chromosome in your partner, and together they form a full pair of chromosomes. And this happens when the sperm meets the egg and fertilization happens. So this is a comparison of mitosis and meiosis. But overall, the PMAT is really similar. Now let's talk a little bit about aging and cell death. So aging is a normal process, as we mentioned a lot of times, and there are cells, as we talked about, that do not divide a lot, and there are cells that divide quite frequently. So for example, cells on your skin are constantly undergoing mitosis because they're worn off, they need replacement. And then there are cells that do not go over mitosis a lot. They're after they're produced, they enter the G0 phase directly and also permanently. So these can be skeletal muscle cells and nerve cells. So they cannot be replaced naturally by the body. Another aspect of aging involves telomeres. So telomeres are DNA sequences found at the tips of chromosomes. So these are basically just replicating sequences of DNA at the tip of chromosomes. They protect the chromosomes from erosion and from sticking onto one another. And as our body cells, as our somatic cells go through cell cycles, these telomeres, they are shortened. The mechanisms are not completely understood, but some studies have suggested that the shortening, the erosion of these telomeres at the ends of DNAs could be associated to aging and cell death. And then we have apoptosis and necrosis. So these are just two, these all refer to the process in which a cell kills itself, you can understand it. So apoptosis is programmed cell death. This can involve lysosomes that we talked about earlier. So after a cell receives signals, for example, or when it is too damaged, it undergoes apoptosis. So this is a normal process. However, necrosis is usually cell death due to pathological causes. So necrosis is not a normal process that we usually see in the body, and basically it is related to pathological causes or otherwise what we call diseases. So so I think this would be it for today. Let's do a quick recap of what we covered in this chapter. So we divided the cell into three main parts. Firstly, we discussed the structure of the plasma membrane, some of its functions and properties. We talked a lot about the three modes of transport across the plasma membrane in terms of energy, concentrations, etc. Next, we talked about the cytoplasm. We talked about what is the cytosol, which are the three parts of the cytoskeleton. And we talked about the organelles that we usually find in a human cell. Next, we spent some time on the nucleus, some of its structures and how it is important as the storage center for our genetic information. We talked about the process of protein synthesis, the two main stages, how it all happens. We talked about this in quite a lot of detail, and I hope you understood the whole process. Next, we talked about cell division. We explained the process of mitosis and meiosis. We distinguished between them, and we talked about why it's important for reproduction. And last, we quickly have gone over aging and the two forms of cell death. So I hope you took away something that is useful from this chapter. I hope you learned something interesting. I hope you enjoyed this video. And if you like the video, please give me a like. You can tell me in the comments if I got something wrong, what you think about the video, any feedback. I'm totally open to that. And if you want further content about the series, about the next chapter, feel free to subscribe. So in the next chapter, in chapter 4, we're going to go another level up. So we talked about the chemical level, we talked about the cellular level. Next, we're going to talk about the tissue level of organization. So if you're interested, stay tuned. And thank you everyone, and see you in the next one.